Welcome to my channel BioQuest MCQs. In this video we will see some important MCQs on metabolism. The electron transport chain is located in the outer mitochondrial membrane, inner mitochondrial membrane, mitochondrial matrix, cytoplasm. Answer is inner mitochondrial membrane. The electron transport chain is embedded in the inner mitochondrial membrane where it facilitates the transfer of electrons and the production of ATP through oxidative phosphorylation. The study of energy changes in biochemical reactions is genetics, biotechnology, biochemical thermodynamics, biochemistry. Correct answer is biochemical thermodynamics. The prosthetic group present in cytochromes is biotin, flavin mononucleotide, iron sulfur cluster, heme. Answer is heme. Cytochromes contain a heme prosthetic group which includes an iron atom that can undergo oxidation and reduction. And this is essential for electron transfer in the electron transport chain. Which of the following vitamins is required for oxidative decarboxylation reactions? Vitamin B1, vitamin B6, vitamin B12, vitamin C. Answer is vitamin B1, that is thiamine. The energy actually available to do work is known as free energy, enthalpy, entropy, biological oxidation. Answer is free energy. Which is defined as the loss of electrons? Reduction, oxidation, electrolysis, electrophoresis. Answer is oxidation. All the enzymes involved in biological oxidation belong to class transferases, ligases, isomerases, oxidoreductases. Answer is oxidoreductases. The degradative processes are concerned with the breakdown of complex molecules to simple ones are called as catabolism, anabolism, amphibolism, metabolism. Answer is catabolism. The biosynthetic reactions involving the formation of complex molecules from simple processes are known as metabolism, amphibolism, anabolism, catabolism. Answer is anabolism. Cyanide inhibits the which site of the electron transport chain? Complex 1, complex 2, complex 3, complex 4. Cyanide inhibits complex 4, that is cytochrome C oxidase site. Cyanide inhibits cytochrome C oxidase, that is complex 4, preventing the transfer of electrons to oxygen. This blocks ATP production and can be fatal due to cellular respiration failure. A more negative redox potential represents a greater tendency to gain electrons, lose electrons, act as an oxidizing agent, accept hydrogen ions. Answer is lose electrons. A more negative redox potential means a substance has a greater tendency to donate electrons, that is, to lose electrons. Such substances are strong reducing agents and they can reduce other compounds by giving away electrons. The bonds responsible for a majority of high energy compounds are hydrogen bonds, peptide bonds, phosphoanhydride bonds, glycosidic bonds. Answer is phosphoanhydride bonds. Phosphoanhydride bonds, like those found between phosphate groups in ATP, they store large amounts of energy. When these bonds are broken, energy is released for cellular processes. A negative sign of free energy, that is delta G less than 0, indicates that the reaction is endergonic, at equilibrium, exergonic, non-spontaneous. Correct answer is exergonic. A negative delta G means the reaction releases free energy and it is spontaneous. That is, it can occur without an input of energy. Such reactions are called exergonic. The storage form of high energy compound in invertebrates is ATP, glycogen, creatine phosphate, arginine phosphate. Answer is arginine phosphate. In invertebrates, arginine phosphate serves as the primary storage form of high energy phosphate. It functions similarly to creatine phosphate in vertebrates, acting as a quick source of energy for ATP regeneration during high energy demands. The main regulatory enzyme of glycolysis is hexokinase, phosphoglucose isomerase, pyruvate kinase, phosphofructokinase 1. Answer is phosphofructokinase 1. In gluconeogenesis, which of the following enzymes bypasses the pyruvate kinase step? PEP carboxykinase, glucose 6 phosphatase, hexokinase, phosphoglucomutase. Answer is PEP carboxykinase. Which coenzyme is required for transamination reactions? Biotin, FAD, NAD+, pyridoxal phosphate. Answer is pyridoxal phosphate, PLP. Superoxide is converted to hydrogen peroxide by the enzyme catalase, peroxidase, superoxide dismutase, glutathione reductase. Answer is superoxide dismutase. Superoxide dismutase catalyzes the conversion of superoxide radicals into hydrogen peroxide and oxygen. And this step is crucial antioxidant defense mechanism in cells to protect against oxidative stress. The end product of anaerobic glycolysis in human muscle is ethanol, lactate, pyruvate, acetyl coenzyme A. Answer is lactate. One of the following components of ETC possesses isoprenoid units: coenzyme Q, cytochrome B, non-heme iron. Coenzyme Q is the correct answer. Coenzyme Q, it is also known as ubiquinone, is a lipid-soluble component of the electron transport chain in mitochondria. It has a long isoprenoid side chain which allows it to move freely within the inner mitochondrial membrane and transfer electrons between complex 1, 2 and complex 3. Cytochromes B, C, etc. and non-heme iron proteins do not contain isoprenoid units. The P:O ratio for the oxidation of FADH2 is 1, 2, 3, 4. Correct answer is 1. The P:O ratio refers to the number of ATP molecules synthesized per atom of oxygen reduced during oxidative phosphorylation. When FADH2 donates electrons to the electron transport chain via complex 2, fewer protons are pumped across the inner mitochondrial membrane compared to NADH. As a result, the oxidation of one FADH2 leads to the synthesis of approximately 1 ATP molecule. Name the compound with the greatest standard free energy of hydrolysis: ATP, phosphocreatine, cyclic AMP, phosphoenol pyruvate. Answer is phosphoenol pyruvate. Phosphoenol pyruvate has the highest standard free energy of hydrolysis, that is 61.9 kilojoules per mole, making it one of the most high energy compounds in metabolism. It donates its phosphate group readily, driving reactions like ATP synthesis in glycolysis. The first step in the urea cycle is the formation of urea, carbamyl phosphate, citrulline, ornithine. Answer is carbamyl phosphate. ATP synthase activity is associated with the mitochondrial enzyme complex 5, 3, 4, 1. Correct answer is complex 5. ATP synthase is also known as complex 5 in the mitochondrial electron transport chain, and it is responsible for synthesizing ATP from ADP and inorganic phosphate using the proton gradient generated by the other complexes such as 1, 3 and 4. Complexes 1, 3 and 4, they are involved in electron transfer and proton pumping but not direct ATP synthesis. Inner mitochondrial membrane is impermeable to Hydrogen ions, potassium ions, hydroxyl ions, all of them. All of them is the answer. So inner mitochondrial membrane is impermeable to hydrogen ions, potassium ions and hydroxyl ions. The inner mitochondrial membrane is highly selective and impermeable to most ions and small charged molecules to maintain the electrochemical gradient which is necessary for ATP synthesis. Hydrogen ions or protons impermeable, their controlled movement through ATP synthase drives ATP production. Potassium ions, they are also impermeable, specific transporters are needed to move them across. And hydroxyl ions, they are also impermeable, their movement would disrupt pH and membrane potential. Which of the following process occurs in the cytoplasm? Citric acid cycle, electron transport chain, glycolysis, beta oxidation of fatty acids. Answer is glycolysis. So the remaining like citric acid cycle or Krebs cycle, then electron transport chain and beta oxidation of fatty acids takes place in mitochondria. So the correct answer is glycolysis. This will takes place in cytoplasm. So thank you for watching my video. Please like, share and subscribe. Thank you very much.